Hello, this is Professor Young, back with you. We're going to look at Chapter 18, all in one fun-filled swoop, going right on through from the basics, right on down to a complete review of, once again, the different types of leukocytes that are present within the white blood cell lineage. And so without further ado, if you would, why don't we go ahead and get started with Chapter 18, the circulatory system. Specifically, we'll be dealing with blood. And what better place than to start with the basics? We should remember that blood is a type of mesenchymal derivative. It is a connective tissue. And again, what makes it that connective tissue is the fact that it does meet all of the requirements that all other connective tissues follow. There is an amorphous ground substance that is present. It does have a main cell type, and in addition to that, it does have a fiber type to it. What is what? Well, if you recall, the amorphous ground substance within blood is plasma. The major cell type are the red blood cells, and of course the major fiber type is fibrin, which comes from fibrinogen, which is the liquefied version basically of fibrin. Fibrinogen, the fibers dissolve within the plasma so that clotting does not happen within the arteries and the veins. And it's not until the contact with the atmosphere, the external atmosphere, that fibrinogen then begins to come back together or reconstitute itself back into the fibers that we therefore call fibrin. And that is basically the groundwork for the scaffolding that will then go ahead and begin to induce healing. It'll go ahead and clot and begin to develop that scab over the open wound. And so, again, blood is that connective tissue. It meets all three of those basic criteria. It has that amorphous ground substance. It does contain a cell type, and it does have a fiber. Functions. The three big functions that we can attribute back to blood are transportation. When we talk about transporting materials, we are talking about the transportation, of course, of oxygen, but not only oxygen, we also have CO2, and we have a whole host of nutrients that become absorbed into the bloodstream to be carried to other areas, specifically nutrients from within the gastrointestinal or the GI tract. As we've just got done looking at in Chapter 17, we know that the blood or the circulatory system will also carry hormones, and it also is a key mover and shaker, if you would, in the movement of heat and waste. So through blood, so when we overheat, heat can be transferred into the circulatory system, brought up into the surface of the skin, into the capillaries, where that heat can be given off in the form of perspiration, sweat, or just the radiating out of the heat. And of course, with that, we can also move waste products. The O2 is a waste product. Just to name one example that can be go ahead and carried through the circulatory system. When it comes to the regulation, of course, the big thing that should be screaming into everyone's head as soon as we hear regulation is homeostasis. Specifically, we are dealing with the homeostasis of pH balance as well as uh, body temperature. And so they're the two big regulatory areas that blood plays a role in. And then there is protection. And when we talk about protections, we're talking about antibodies, and that would be antibodies through the white blood cell lineage. White blood cells themselves forms a type of protection, and then, as we know, with clotting, or I should say with fibrinogen and fibrin, we have the clotting factor, which does serve as a protective measure. Otherwise, you'd bleed out and, expire. Just a couple more words about the basics here, or an overall introduction into blood. Blood is slightly alkaline. In other words, it's above that 7 threshold, and it's just barely above 7. We're not talking about too far off neutral, but it does like to be alkaline and not acidic. And so that pH typically lies or varies between 7.35 and 7.45. Not a huge difference there. The color of blood is going to vary dependent on oxygen. When we have oxygen-rich blood, the blood appears to be bright red. And then what happens is, as we lose oxygen content from within the blood starts to become a darker shade of red, which very often appears to be blue when we look at it through the skin. It's less oxygenated blood. Blood is always red. We don't magically turn colors with our blood. It doesn't go from red to blue. I don't know of any critter that does has two completely different colors to their blood. There are different colors of blood, as we discussed within in lab. Horseshoe crabs contain green blood, and there was another example that I gave that's slipping my mind right now. But at any rate, the body contains between 4 and 6 blood, and it likes to have that key temperature in and around 104 degrees Fahrenheit. Again, that's to help maintain overall body temperature. What is this thing that we call blood? Well, it has two key areas to it. The first area is liquid base, which you can tell from our diagram right here, that the majority of blood, over half of it, when we say over half of it, of course, we're referring to, as you can see, 58% of blood is plasma. It is that liquid foundation to it. The remainder of that blood, that other 42%, is made up of a couple different things. The first thing it is made up of is of course, red blood cells, which would be the second most significant part of that. That 99% of the formed elements, or 99% of the other 42%, is red blood cells. And then we have a fine line, if we would. We have a fine line for our white blood cells, which tends to fall right in there. Why didn't that come up? There it goes. Which falls right in there. And that's our white blood cells, which is less than 1%. And then your platelets are even smaller than that. Remember, platelets are simply broken up particles found within the blood, typically of older cells that have gone on to a better life as platelets. Blood plasma, again, 58% of your blood is made up of this blood plasma. Of that 58, or of that plasma, about 91.5% of it is water. And so when you become dehydrated, your plasma loses water content, which means it becomes thicker, which means those red blood cells and white blood cells and platelets have a harder time passing through the circulatory system, because now the viscosity, that might be a new term for you, viscosity is the ability to flow. And the higher the viscosity, the thicker the fluid is, the harder of a time it will have to flow. When we think of things that have high viscosity, maple syrup frozen in the freezer has a very high viscosity, whereas water has a very low viscosity. And so we like to have a high content of water because it lowers the viscosity, it lowers the resistance to movement, and so that keeps the blood flowing. And then the other 8.5% or so are mainly proteins that help to, again, provide nutrients within that blood. And you can see some information there. Proteins are synthesized within the liver, specifically within a type of cell that we call hepatocytes. And what they are doing, or I should say the major proteins that are formed, are albumin, globulins, and fibrinogen. There we go again. Of course, the fibrinogen is dissolved within the blood to form fibrin, which is used within clotting. Albumin is going to be a transporter for steroids, and then the globulins, things like antibodies and transportation of iron and all that kind of good stuff there. The formed elements, of course, we have the red blood cells, which we will talk about here in a few minutes, as far as how the red blood cells are produced. But the red blood cells make up the majority of the formed elements. And then we have white blood cells, for which we spent quite a bit of time the last class really looking at and trying to hone in your ability to differentiate the different types of white blood cells. There are 5 main types. You have your neutrophils, your lymphocytes, your monocytes, your eosinophils, and your basophils. Don't worry, we'll go through that again here. And then your fragmented cell types, and that being your platelets. So that's what's in your formed elements. Your red blood cells, I'm going to talk a little bit about that before we move on to our leukocytes, or our white blood cell types. So red blood cells, hematocrit, which is something that they measure. If you ever look at a blood report that you get from your doctor's office or from your hospital, you will see the hematocrit. And what that is basically talking about is what is the percentage of red blood cells making up your blood. We know that it should be near to about 48% or so, depending on the sex. Females tend to have a little bit lower of a red blood cell count within their blood, typically 38 to 46%, whereas males will have 40 to 54% red blood cells within theirs. Now keep in mind, what allows males to have that higher percentage or that higher expectation of a greater percentage of red blood cells is the fact that testosterone is the hormone that will trigger or stimulate the whole process of hematopoiesis, or the formation of red blood cells. And it does that by testosterone itself synthesizes the hormone erythropoietin, EPO, and it's that hormone that will then go into the red bone marrow and begin or spark, if you would, the production of red blood cells. And you can see typically about every second or so, we have about 2 million new red blood cells entering into our circulatory system. And so, yes, this definitely, without a doubt, is a continuous process. It is a very expensive process because of how we have to consistently replenish the supply of red blood cells. We know that at least from the perspective of the old fogies, that being the adults, that red blood cell or hematopoiesis occurs within red bone marrow. And we find the red bone marrow within the ends of our long bones, but we also find red bone marrow, trabeculae, and all that kind of good stuff within the flat bones, within the skull, within the scapula, within the rib cage and the sternum, within the pelvic area. But that's not how it always is all through development. The location of red blood cell production shifts with age. And so when you are a fetus developing in your mama's womb, such as in this area, it is not your bone, pay attention to this, it is not your bone that is producing red blood cells. Why would your bone, while you are a developing fetus, not be producing red blood cells? Why does your body as a fetus rely on the liver, the spleen, the thymus, and the lymph nodes? Why not just have the bones do it? Yes, I'm expecting you to answer this. I'm expecting you to go ahead and talk out loud to your computer screen. I think someone said, if you didn't say this, say it as soon as I'm done saying it. The reason why fetuses do not rely on bone, red spongy bone, for the production of red blood cells is because they don't have bone. They've got cartilage. Don't get tripped on that one. I'm warning you now. All right, so the fetuses are going to rely on the liver, the spleen, the thymus, and the lymph nodes for their red blood cell production. Once they have graced the world with their appearance as a infant or a child or an adolescent, then we see that switch. And that it's a maintained switchover, if you would, from the liver, the spleen, the thymus, and the lymph nodes to red bone marrow producing red blood cells. Now, notice I didn't say necessarily spongy bone. Notice I didn't say flat bones. Notice I didn't say the ends of the joint. I said as a adolescent, as a child, or as a infant. Red bone marrow is what is producing the red blood cells. Your body has red bone marrow present everywhere in the body except the front of the face. And so the entire body of a child, of an adolescent, of an infant is producing red blood cells. It's not until around age 8 or so that we begin to see that switch over, even within the long bones, especially within the long bones. We don't see that switch over until in and around age 8 for red blood, or the not red blood cells, red bone marrow switching over to yellow bone marrow within the long bones, specifically the diaphysis of those long bones. Again, that happens in and around age 8. The red bone marrow begins to switch over to yellow bone marrow because of the presence of the adipocytes. And so it goes from being a production center for red blood cells to an energy storage facility, hence the main cell type being adipocytes. Now, if you thought that was fun, let's spend a little bit of time talking about how exactly we produce these red blood cells. This is a multi-step process. And what I need you to desperately understand is that It's almost like one of those decoder games or mystery games. If A happens, then B, C, D, E, or F could happen. If B happens, then we jump to K. If C or D happens, then we jump to L. But if E happens, we jump to M. And so it's kind of one of these games that we have to play, and it becomes very convoluted when you're trying to understand this. So follow with me just for a little bit. When we are making red blood cells, and this goes all the way back to the red bone marrow. So we're back into the bone. Yes, we talked about skeletal system already back in AMP1, but we're back in the bone again because this is the hub. This is the grand central station for red blood cell production. We've got these stem cells, these undifferentiated cells found within the trabeculae in the spongy bone called pluripotent stem cells. These pluripotent stem cells, and I'm going to try to color code this as much as I can for you. So these pluripotent stem cells have the ability to differentiate into a host of different cells. They can become red blood cells, which is what we are interested in. But in addition to that, they can also become macrophages. They can also become reticular cells. They can also become mast cells. They can also become adipocytes. They can also become osteoblasts. They can also become chondrocytes, or chondroblasts, sorry. They can also become muscle cells. So we're not just locked in to these pluripotent stem cells strictly becoming red blood cells. Pluripotent stem cells will differentiate sometimes into red blood. Sometimes they will start the production process for red blood cells. Sometimes they will differentiate and start the process of becoming macrophages. Sometimes the pluripotent stem cells will differentiate and start the process of becoming reticular cells, or start the process of becoming mast cells, or start the process of becoming adipocytes, or osteoblasts, or chondroblasts, or muscle cells. We are not interested right now in anything in here. The only thing we are concerned about are these guys, the red blood cells. That is all that we are concerned about right now. And so you need to understand that while all of this other stuff is happening, we're kind of wading through and pinpointing exactly the process of the red blood cells. So these pluripotent stem cells will go ahead and will differentiate into a lineage of cells that will eventually become red blood cells. Specifically, what are they? What cells will they differentiate into to become red blood cells? Well, this answer lies with specifically the myeloid cells. Right there. That's the cell type we want to focus in on. Right. Before that happens, we have to give a little bit of due recognition to our reticular cells. Because what happens here is the reticular cells will begin to form a network of fibers within the spongy bone. And those reticular cells will serve as the stroma, or the bedrock, if you would, for red bone marrow cells to be produced. Once the red bone marrow cells have a place to anchor onto, hence the reticular cells, then they can focus on producing the red blood cells. so that the myeloid cells have a place to go ahead and go to, if you would, once they've differentiated from the pluripotent cells. Right, so that's the first thing that I need you to understand. Pluripotent cells, in order to create the lineage of red blood cells, will differentiate into myeloid cells, but pluripotent cells will also differentiate into a lineage of cells that will become reticular cells. The reticular cells are especially important because that serves as an anchoring for red bone marrow cells. Got it? Good. The myeloid cells, come back to the myeloid cells real quick. They form within the red bone marrow, as we just got done talking about, and they will divide to start to continue along the process of becoming red blood cells. All right, now, let me go ahead and clear the board here. Myeloid cells that are going to continue along the journey of becoming red blood cells will divide and become so the myeloid cells here will divide and become progenitor cells. So we're going from the myeloid cells to progenitor cells. Once the myeloid cells become progenitor cells, they are locked in on a path. The progenitor cells have to commit in becoming the myeloid cells, or the progenitor cells have to commit to becoming either red blood cells, platelets, I'm missing an E in platelets there, I realize that, or they will commit into becoming neutrophils, or they will commit to becoming monocytes. So the progenitor cells will then begin to divide into and continuing along the lineage of either becoming red blood cells, platelets, neutrophils, or monocytes. Following me? I hope. Once they decided where they're going to go, they can't divide and become anything else. They're going to be committed to becoming one of these. So if the progenitor cells differentiate into precursor cells, if the progenitor cells differentiate and become precursor cells, they have locked in their fate into becoming red blood cells. Don't worry about platelets anymore. Don't worry about neutrophils anymore. Don't worry about monocytes anymore. Once the progenitor cells have differentiated into the precursor cells, they've locked their destiny into becoming red blood cells. They have no choice. Precursors will undoubtedly become red blood cells. This is what happens. Prourethroblast will begin to divide within these precursor cells to become and create hemoglobin. We'll talk about hemoglobin in a couple minutes. But we know from lecture that hemoglobin is the molecule that is attached to the red blood cells that, because of the hemi group, will go ahead and carry the oxygen. Towards the end of the process of the prourethroblast creating hemoglobin, the precursor cells begin to lose their nucleus. And it is at that point in time when it goes from being a erythroblast to a reticulocyte. Once the hemoglobin is formed and the nucleus disappears, you go from being a proerythroblast to a reticulocyte. With the reticulocyte, the soon-to-be red blood cell continues to develop. With the nucleus out of the picture, the reticulocyte becomes this biconcave structure. It becomes this biconcave structure. That typical image that you think and hear about when we talk about red blood cells. It'll keep some of its mitochondria. It'll keep some of the ribosomes. It'll keep some of the endoplasmic reticulum. Not a whole lot of it, though, but it will keep some. At that point in time, no more nucleus, no more proerythroblast. It is a reticulocyte. It is biconcave. It's got its hemoglobin. It's still inside of the red bone marrow, and it goes bon voyage. It sets sail. It sets sail into the plasma, into the blood, and circulates through the body. After about 2 days of circulating through the body, you have yourself that mature, fully carrying red blood cell. And so that's the progression that we need to look at. Again, the control goes back to these hematopoietic growth factors. Erythropoietin that we talked about before, EPO. It is what leads to the direct stimulation of the erythrocytes, aka blood cells, red blood cells. That's all erythrocytes really stand for. Are red blood cells. And then thrombopoietin, TPO, would direct the formation of the platelets. Now, hold on 1 second, if you would. All right, sorry about that. So the structure of red blood cells. We've already talked about this a little bit. We kind of know about this. We know that red blood cells, and here's the key thing that I want to point out on this particular slide, and that is red blood cells themselves cannot reproduce. And this is why a couple minutes ago I made mention that this is an expensive process. Because once these red blood cells are produced, they're not dividing on their own anymore. It's got to go back to the red bone marrow to be able to produce more red blood cells. And so that's a key thing here for us. All right. The structure of hemoglobin, we mentioned hemoglobin briefly earlier. We have two major areas. We have the globin and we have the hemi. The globin would be the protein chains. You have the alpha chains and the beta chains. It is within these globin chains where we see carbon dioxide being stored and carried throughout. It is the hemi, and the hemi here are these orange little pineapple-looking discs. We've got 1 and 2 and 3 and 4. We've got those 4 discs right there. They are the oxygen-carrying molecules. They've got iron in the center that attracts the oxygen and then binds it on. What is key here, and what I want to make sure that you understand, hold on 1 second, is that each red blood cell will go ahead and basically store, and we figured this out in class, some 280 million hemoglobin molecules. Each hemoglobin molecule can go ahead and hold 4 oxygens. So this was something. It was something in the order of 1.1 million oxygen molecules could be transported throughout the body at any one point in given time for one cell, for one red blood cell. So keep that in mind. Each red blood cell can go ahead and carry about 1.1 million oxygen molecules throughout the body. And if that doesn't startle you, then I don't quite know what will. All right. White blood cells. We talked again about this a little bit more. These slides are going to pop up again on here. I want to make sure that you understand what it is that you are looking at so that you are better prepared both for my lecture exam and as well as the looming lab practical. Slang for white blood cells, if there was physiological street slang, would be the leukocytes. They do contain a nucleus, unlike their counterparts, the red blood cells. They do not contain hemoglobin like their counterparts, the red blood cells. And we see that these 5 types of leukocytes are broken up into 2 types, granular and agranular. Granular, it appears to be very pixelated at times, whereas the agranular are more solid, more well-defined regions. And so if we look at this slide here, that's slowly coming up. There we go. We see what these 5 leukocytes look like. The neutrophils. We'll start with the neutrophils. We're going to outline them guys in red. The neutrophils are basically present for bacterial infection. They are pus producers. They help in inflammation, which is why typically wounds get very red and swollen. It has to do with the presence of the neutrophils. They are very characteristic in their appearance in that they are generally either S-shaped or C-shaped. And you can see the one there that we are looking at in front of you right now is that C-shaped. They also tend to have anywhere between 3 and 5 lobes contained within that nucleus. And you can clearly see that, at least I think you can, with this guy here. You've got 1, you've got 2, you've got 3, and you've got 4. And so there's 4 lobes present there, typically between 3 and 5 for the neutrophils. And these guys account for about 60 to 70% of the white blood cells present within the body. Next, we draw our attention to the eosinophils. These guys right here, we'll label them in blue. Probably among my more favorites just because of what they help to combat. Usually we see these guys present within parasitic infections. So if you have a parasite inside of you, you're going to have these guys present. And you also see them with allergy and asthma spikes as well. When you look at these guys, they typically have two larger areas for the nuclei. And yes, they can be rather pixelated, but you can typically make out still the nuclei or the nucleus that is present. Next, we turn our attention to the basophils. With the basophils, we see these in response to allergens once again. We see these in response to allergens, and typically they will go ahead and release histamines and or heparin to allow the blood to flow. That is one of the things that we would like. Heparin being a blood thinner and histamine acting as a vasodilator. Typically, you only see about 0.5% to 1% of the white blood cells in your body at any point in given time are basophils. One of the things I do want to point out to you is typically the basophils are so pixelated that the nucleus is unseen because the cell itself is dark and again has that pixelated appearance to it. Next, we're going to jump over to the lymphocytes. We're going to leave the monocytes for last. Lymphocytes are going to be present for fighting infections. These guys are also the cancer destroyers. If I can spell destroyer, that would be really good. Cancer fighters, there you go. And so typically if someone has cancer, their lymphocyte count will be much, much higher. The other thing these guys do is they will summon other white blood cells that might be needed to kind of come in. As the macrophage lineage, it will come in and basically break down any kind of debris. So the lymphocytes will attack the cancer cells and then signal for the monocytes to come in and be the street sweepers and clean everything up all nice and neat. Typically, because these guys have a role in fighting infection and because they also have a role in summoning, forget about it, other types of white blood cells, such as the neutrophils and the basophils and the monocytes. We see a little bit higher percentage of these guys making up the white blood cell count, and so that would be about 25 to 33%. And then last but not least, bringing up the ugly green color, we have monocytes. Again, these guys are in charge of phagocytosis or breaking down kind of other kinds of dead neutrophils or dead pathogens. And again, just kind of cleaning things up. They tend to be kidney-shaped. In appearance. And so these are your 5 major classes of white blood cells and or physiologically street lingo would be the leukocytes. And so how good are you? That is the question. You should be able to identify, or at least start to identify, some of these guys and who they are and what they are doing. If you cannot, we need to go ahead and get you there, so that this guy that is located right here, we notice that there is no clearly defined nucleus, but it is highly pixelated, and so therefore, without much left to really consider, you should be able to identify that as a basophil, hopefully. This guy here, we're going to go with 1 first. That 1 there has that characteristic C shape, and so you should be screaming so that everyone around you can hear you, that is a neutrophil, whereas this guy over here, who we will aptly call number 2. Number 2 would be a what? Large, dark nucleus taking up almost the whole cell. I think I heard someone say lymphocyte, and if you did, congratulations, you are correct. There are, just looking at this slide here, there's a couple little guys here, there. That very well could be platelets. Of course, they could be, I would gather and put my reputational lenses, they are platelets there. This next one here, 1 and 2, they are both the same. So 1 and 2 are both the same type, kind of an S-shaped, kind of a C-shaped. You've got definitive lobes. You've got 1, 2, and 3. 1, 2, 3 there as well. And so therefore we would consider these S-shaped or C-shaped multi-lobed leukocytes to be neutrophils. Then we have this guy here, 2 very clearly defined nuclei. You've got an area that is, these 2 nuclei, apparent nuclei, are attached by a very thin strand right there. And so by definition, we have no choice but to classify these as what? Eosinophils. Very good. And so if you are on top of that, that is what you should have labeled that. Hopefully. For Valentine's Day, Valentine's Day can be any day of the year. It doesn't have to be in the month of February. And so we have our heart-shaped leukocyte. There is some debate on this, and I don't do this often, but I'm actually going to change what I thought this could have been. I'm now going to define this as a monocyte. It is kidney-shaped. It is very darkly stained. They are not 2 separate nuclei, and that is a monocyte. And then we've got the fearsome 8. Again, number 1 was the neutrophil. Number 2 was a neutrophil. Number 3 were the platelets. Number 4 was a lymphocyte. Number 5 was a lymphocyte. Number 6 was a basophil, and number 7 was a neutrophil. And so you've got multiple there. By the way, this is a type of bone cancer, if I'm not mistaken. if I remember correctly. You can't really tell from that, but I just remember that being the tagline on this one. And here again we have there two cells. Cell number one, which is right over here, is a lymphocyte. And then cell number two, which is the lower one, is the, if you said eosinophil, you are correct. That was just an evil trick. That's a follicular cell. We're not going to worry about that right now. Here we have all what kind? Large, darkly stained nuclei. Lymphocytes. Very good. Last little bit that I want to touch on, and then I promise I'm going to leave you alone, at least until you have questions in lecture or until your lecture exam. Real quick, just on some blood typing and that kind of stuff. The ABO blood groups are differentiated based on antigens that are found on their outer surface. Those antigens that can be present on the surface of a red blood cells are called agglutinogens, and those agglutinogens basically tag the red blood cell. And so what they're tagging it against is either the A or the B antigen. It won't attack oneself, and that's the key thing there. So the antibodies would detect the foreign antigens and destroy them. And so if you are a B blood type, you would have B antigens. If the antibodies detected A antigens, it would go ahead and would attack those blood cells and basically destroy them. The antibodies that we are specifically referring to here are the agglutinins. And so you have basically, like, for if you are a B, if you are blood type B, then you would have anti-A antigens, and that would basically be, it's not going to look pretty. Hold on one second. Anti-A antigens, or what is sometimes also referred to, and you don't usually hear it as this, but just in case you ever do, alpha agglutinin is what it would be referred to, and that would be anti-A. If you are blood type A, then you would have anti-B or beta agglutinin, and that would be if you have that blood type A. AB is universal, and the reason why AB is universal is because it has both A antigens and B antigens, and so it won't attack itself no matter what. And blood type O, which is what I am O positive, doesn't have any antigens at all, and so we are a universal donor to anybody, because our blood can go into an A and not be detected because it doesn't have any antigen markings on the surface. And it can also do that for the Bs as well. This here is just simply a real quick reminder. The A agglutinins only for the red blood cell, you can see, is tagged with the A antigens. Blood type B, whether it's BB or BO, is marked with the B antigens. Type A is marked with both A and B. Type O has no agglutinogens on them. And the plasma antibodies that are there, again, if you're type A, then you've got type B antibodies. If you're type B, then you have type A antibodies. If you're type AB, you have no antibodies, and if you're type O, then you've got A and B antibodies, which is why type O's can only give the type O's. I'm sorry, type O's can only receive type O's because they do have the A and B antigens, but they can give the A and B because it doesn't have any surface proteins on them. And lastly, and I know this has been a long one, guys, and you're hanging in wonderfully, and I appreciate that. We have the RH blood groups, or the RH factors. I will note that RH, the RH factor, RH positive, RH negative, the RH comes from the rhesus monkey. It's not REESES. That would be one of my favorite candies. That would not be the monkey that we are referring to here. But the rhesus monkey, RHESUS, is what was the animal that was studied and used to determine and discover the RH factors. And so you'll notice that, you know, if someone's I'm O, I already said I was O, type O positive. Type O, the O there would indicate the blood type. That positive would indicate the RH factor. And there's basically three different types of antigens that are associated with the RH group, C, D, and E. And what the positive and negative represent is whether or not the most reactive of those antigens, antigen D, is present or not present. If you are RH positive, then you have the D antigen. But if you're RH negative, then you're lacking that antigen. And really, this only comes into play when the person is subjected to the opposite RH factor. And so it's really independent, if you would, of the blood typing itself. And so I would not be able to receive O negative blood because O negative would be lacking that D antigen, and my body would be able to recognize that. And then, actually, no, I can receive O negative. O negative is the universal donor because it is lacking in it. Where it would come into play is if a RH negative person came in contact with an RH positive person. That's what it would be. That would be the issue. So if I was O negative, I could not receive O positive blood because of that RH factor being positive. And just to demonstrate this a little bit, here is a nice little reminder. The mom here is RH negative. The developing fetus is RH positive. During birth, there is ripping and tearing and everything else that goes on during delivery. It's not a big deal for the first child. It's not a big deal for the first child because what happens is the child gets out okay, and the mother now has the antibodies, if you would, to go ahead and recognize that foreign body as being foreign as blood mixes. And so as that baby is leaving, there's ripping, there's tearing, there's blood smearing and blood sharing and everything else that happens during that wonderful, magical process of childbirth. But the child gets out. The positives are out, but the positives are picked up again within the mother's blood system as being there, and so they develop those little antigens. That's what those little blue things are there. There's the antigens. During the second pregnancy, the umbilical cord is attached to the fetus, and there is blood exchange that happens periodically sometimes throughout that, especially if there's ripping and tearing within the umbilical cord. The antigens are already there for that positive RH factor, and literally the mother's body begins to attack the fetal body with those with the antigens, and so there's a shot that basically blocks the binding sites, and that shot, if I'm remembering correctly, I think is referred to as the RH immune globulin. something along that lines. That's the kind of what they give the mother to go ahead and block the binding sites, if you would, so that the antibodies don't attack the fetal, the developing fetus. So there you have it. 27 slides, probably darn where's near 45 minutes to an hour or so of Chapter 18. Hopefully you find this beneficial. As always, feel free to rewind, pause, really study, look, try to concentrate on what's going on with this. If you have any questions, feel free to email me or jot them down, and you can ask them during the next lecture session that we have together. Until then, have a good day, happy studying, and this is Professor Young signing off.